This video, from the Institute of Agriculture and Natural Sciences at Deen Dayal Upadhyay Gorakhpur University, focuses on the general introduction, classification, and breeding methods of the brinjal crop. I am Anurag Kushwaha, a first semester MSc Ag Horticulture student. Brinjal, or eggplant, is scientifically known as Solanum melongena and belongs to the Solanaceae family. It has a chromosome number of 2n = 24 and originated in India. It is primarily a self-pollinated crop. Some varieties are called eggplant because their white, oval fruits resemble chicken eggs. Brinjal is a principal vegetable worldwide and possesses medicinal properties. It is rich in vitamin C, vitamin K, folate, manganese, and potassium. Fried brinjal fruit with sesame oil is an excellent remedy for liver complaints. India is the largest global producer of brinjal, with an estimated production of around 12.9 million metric tons in 2025. It is cultivated across approximately 7 to 8 lakh hectares in India, with top states being West Bengal, Odisha, Bihar, Andhra Pradesh, Karnataka, and Uttar Pradesh. Other major producers include China, Egypt, Turkey, and Indonesia. This chart illustrates the statewide share of brinjal production in India. Global eggplant production is dominated by India and China. In 2025, the top 5 producers are India with 12.9 million metric tons, China with 11.5 million metric tons, Egypt with 1.3 million metric tons, Turkey with 0.8 million metric tons, and Indonesia with 0.7 million metric tons. Brinjal is a warm season crop. The optimal temperature for growth is 21 to 27 degrees Celsius, and temperatures below 15 degrees Celsius adversely affect growth. Seedlings are best transplanted around 20 degrees Celsius. Brinjal can be grown in various soils, but light, well-drained loam or sandy loam rich in organic matter is preferred, with an optimum pH of 5.5 to 6.0. Transplanting is the common method for crop establishment. The seed rate is 500 to 750 grams for open pollinated cultivars and 250 grams for hybrids. Sowing seasons vary by region and season. Seedlings, 25 to 30 days old, are transplanted in the field with specific spacing for non-spreading and spreading varieties. The recommended dose of fertilizers is 100 to 120 kilograms per hectare of nitrogen and 50 to 60 kilograms per hectare each of phosphorus and potassium. Irrigation is given immediately after transplanting and subsequently every 10 to 15 days in cool weather or 4 to 6 days during summer. Fruits are ready for harvesting when fully developed, tender, bright, and glossy. This usually happens 50 to 60 days after transplanting, with harvesting occurring every 5 to 7 days. The average yield is 250 to 500 quintals per hectare, with F1 hybrids yielding 400 to 6,000 quintals per hectare. Fruits can be stored for 7 to 10 days at 7 to 10 degrees Celsius and 85 to 90% relative humidity. Major diseases affecting brinjal include damping off, alternaria leaf spot, bacterial wilt, mycoplasma diseases, and mosaic disease. Common insect pests are the brinjal shoot and fruit borer and the hada beetle. Related species of brinjal include Solanum cicimbrifolium, which is an ancestral form, S. ethiopicum, S. macrocarpum, S. indicum, S. trilobatum, S. vearum, S. nigrum, and S. xanthocarpum are also related, with the latter species used in native medicine. Brinjal has 3 main botanical varieties: S. melongena variety esculentum, which bears round fruits; S. melongena variety serpentium, which bears long fruits; and S. melongena variety depressum, characterized by its dwarf plant stature. In the floral biology of brinjal, the corolla or petals are showy and purple, and there are 5 calyx or sepals. Anthers are bilobed, dark yellow, and attached to the base of the petals. Flowers generally open between 6 a.m. and 8 a.m. in tropical climates, with anthers dehiscing an hour later. The stigma is receptive a day before the flower opens and on the day of anthesis. Heterostyly is the condition where styles have different lengths relative to the stamens in different individual plants, which reduces self-fertilization. There are 3 types of flowers: long-styled with a big ovary, medium-styled with a medium ovary, and pseudo-styled with a rudimentary ovary. Plant breeding involves manipulating plant species to achieve desired genotypes and phenotypes through selection and controlled mating over generations. Key breeding methods include pure line selection, pedigree method, heterosis breeding, and mutation breeding. The objectives of Brinjal breeding are to develop high-yielding varieties, improve fruit color, shape, size, and nutritional value, achieve resistance to insect pests, especially the shoot and fruit borer, develop abiotic resistance to factors like drought and salinity, and reduce seed proportion. Pure line selection involves individually selecting and harvesting a large number of plants from a self-pollinated crop. The best individual plant progeny is released as a pure line variety with an identical genotype. This method applies to local cultivars and land races. Examples include Arca Shirish and Co1. The pedigree method is a technique where individual plants from segregating generations are selected and documented. This aims to develop genetically uniform pure line varieties by selecting plants until progenies show no further genetic variation. It involves selection after hybridization between superior cultivars. Arcanidi is an example. Heterosis breeding utilizes F1 hybrids, which are popular globally and gaining popularity in India. Examples of hybrids include Arca Anand, Arca Nilkanth, Pusa Anmol, and Arca Navneet. Mutation breeding, or variation breeding, involves exposing seeds or plant tissues to agents to induce new traits not present in the original population. PKM1, developed from Pujudi Kathari, is an example of a variety resulting from mutation breeding. Thank you for watching this video on Brinjal breeding.